Class is starting, and the first thing to mention is the pre-med club is up and charging ahead and having its first meeting this Thursday, and that's today, and it's at 5 today. So if you're interested in that, you can go. If you can't make it to it, you can still join the club, and you just have to scan that code. Then the next thing to say is that a couple of students got the wrong ebook, and I contacted the publisher. The publisher doesn't sell the ebook. They have suppliers that sell it, and so communicate with the supplier about shifting your payment to the correct book so you get access to the correct textbook. Everybody remember that if you don't have the book quite yet, chapters 1 and 7 are an electronic reserve. That's on our Brightspace, and pull down the content on our course Brightspace, and that's where the syllabus is, and we'll be there all year and all semester, including a video to explain the course. And also, if you go down that list a little bit further, that's where it's got the electronic reserve. And that's the only thing I plan to add to electronic reserve, I think, so, but that's where those two first chapters are. So everybody's rolling forward with that, that's great. Just want to mention again this idea that a bunch of USM students participated in this research internship that is content very similar to this course, studying biomedical topics down at that molecular genetic level. Then, this might be about the last time I talk about this. The requirement for participating in our course is to either sign the attendance sheet or within a week of the course, send me email with pictures of the notes you took following along to the recorded lecture. And now we are in a position where everybody's joined the course. It's going to join, and so one reason I have this kind of set up is so if a person joins a course, my course is late, they can catch up. They didn't miss anything, right? They didn't really miss the classes. And but I'm going to intend to start deleting videos after a week. So the intention is to do our work, do our learning on an ongoing basis. We can't crunch it in just for an exam because it's going to stack up, okay? So there's that. Then what you do this week, and we talked about this, it's the same list, it's just rearranged a little bit because we took our first quiz. And we'll have our second quiz at the end of class today. We'll do an in-class assignment. And as you hand in your second quiz, I have your first quiz up here, and you can pick it up. And so get in the habit of writing down questions, bring them to class. We'll have time for questions. We have at any time. We'll intentionally have time in just a couple minutes. Then we'll do that assignment in class today. We'll have quiz 2 at the end of class about the muscle differentiation videos. For us, they're really clay model videos. We really don't care much about muscle differentiation in this course. We care about the clay models. And they're clay because we can manipulate the DNA because it's physical. It's in a test tube. When we purify it, we deal with it. So chapter 1, then learn by recalling. So I'll talk about that when I show you the next thing. Because we have another thing to add, which is, if you haven't taken a class from me before, when I grade quizzes or exams, I rarely write down comments on them. I rarely write down great or no, that's wrong for this reason. I just take points off like that. And so you're like, well, how will I know? And the purpose of having the quiz is to help you learn. So what happens is I grade the quiz or exam, and then I immediately make a video of myself taking the quiz. And I just post that, so I'll post that today. And in it, I'm trying to help everyone. Because really any comment to any specific student is likely to be helpful to everyone. Lots and lots of times I give full credit for an answer because it deserves full credit, but it could be a better answer, and it should be a better answer, and we should try to build toward that. And so after exams, I often require, as an assignment, students go through the answer key video because I make them pretty fast, but I consider it a pretty significant resource. One way to understand that is in assessing students ever. There are only 2 assessment approaches or purposes, and one is called formative assessment, and the other is called summative assessment. And these quizzes are formative assessment. It's checking in, how's it going? And I want to give you feedback. And then a summative assessment is, this is the end. It's an exam, and here's, show us where your learning is at. So this is formative assessment. It's not worth a bunch of points, and The points are there just to motivate you to participate, really. But it's really to get some feedback like that. And as part of it, for example, I try to coach students about how to answer the questions. So our exams are going to be 50 questions. It's going to be a lot of writing, and I write the exam so you can finish it. I've given a lot of exams, and students aren't really pressured. Like I only got halfway through. It's not like that at all, because that's useless, right? But students have to be efficient, and so like on this quiz, right? One of the questions was, is RNA interference knockout or knockdown? You could just circle one of those. I'm not asking for an explanation or a definition or examples. If I was, I would ask about it. And here's what comes up. There are sort of two answers. And one answer is a short, direct answer, like that definition of orthologs. Two genes in different species that are interested to share a common ancestral gene. Just kind of spit that out. The regulatory region of a gene regulates where, when, and how much of the gene gets expressed. They're just like little memory things. But there's always this second answer possible, and that is, when we're learning about these topics, we could talk about the short definition, but we also then talk about explaining it, putting it in context, giving examples, because it's new to you, and you're trying to grapple with its, meaning. What does this mean, You got to understand it, not just memorize it. And as a result, students want to give one or the other answer on a quiz or exam. Kind of a longer answer with examples, and that's fine. Longer answers are fine. But just recognize that you want to get to the point like that. Yep. And you want to use good words, because it's your answer. And I'm reading it, and I'm like, they don't seem to understand this, right? Like, one thing that's really common for students throughout colleges, they just seem to replace the word gene with cell. And those are totally, that's totally different things, right? Like that's like saying replacing tire and car. And, and I'm sensitive to it and, and recognize it, but I can't give somebody credit if they're like, something about, they mean the car, the people get in the tire. And I'm like, you don't seem to understand it. Right? And I'm not fussy, I'm not grading on grammar, except that it's true that some grammar is required to actually being able to express your knowledge, okay? But I'm not really picky and really fussy and really mean. I'm just like, I got to judge it, right? Does it seem like that student answer seems to understand the thing, right? So sometimes it's good to go with a little bit longer answer because you understand it. You understand the case context of examples and stuff like that. That's fine. But just recognize that these are kind of short answers, and they do have the potential to be this like that. Because this is this kind of car mechanic thing where we're talking about tires, Another thing about it is I'm not a jerk. So, when I'm talking to you, I'm not, looking down at you and being like, you don't get this, right? I've taught these topics a lot, right? And I'm sensitive that, you're doing great, and yet you're struggling to sort of ask the question and explain things. As the students were handing in a quiz on Tuesday, the student was just putting their quiz down and asked me a question about the wording choice they used, and I was like, oh my gosh, it's not a really significant science question, but it was a really good question about like the word, how to use these words. And I actually laid in bed for a couple of hours just thinking about that, and thinking about that. Because one thing that's true is for molecular biologists are especially specific in word choice. Because it's just fact. They want to be really careful and use the right words. And again, I'm not asking you to be, a professor of molecular biology like that, right? I'm very sensitive to it, but just recognize that we're going toward this thing that is just kind of like a bunch of matter-of-fact things, like that. So anyway, watch that because and I'll post that with our link for our class today. Okay. Then we're going to do this homework assignment because I'm trying to encourage you to realize that when you draw this out, you can help understand it more. So that means that sometimes on the exam, I'll say draw it out and label it. Other times I won't say draw it out, but always. If you have a combination of drawing and writing, that's a good answer, right? And so that's part of it too, is you can use some drawings, right? There's a lot of drawing going on here. And we're going to do some more of that. today. And what we did on Monday, let me just see, I think we gotta stop here and we'll talk, and then we'll do some more drawing, because we want a little bit more drawing done. So do people have any questions about anything where we're at? And one thing you might want to ask about is, well, we're having a quiz at the end of class today, which again is going to be at 10:30, and we want to be ready to stop right then. So, you have, maybe that clock's a little bit off or anything, but you have your phone there, you can set your phone. Everybody should set their phone for like 10:30, and that'll be funny, because they're ding, 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 you'll be like, oh, what's that about? The cookies are done. Yes?
My question is, where does the original MyoD protein come from?
Yep, where does the original MyoD protein come from? Yep, so it's a protein. And so whenever there's a protein, there's a gene that codes for that protein. So there's a MyoD protein, there must be a MyoD gene. And the MyoD gene is called a coding gene because it codes for a protein. So when it gets expressed, the transcription unit gets transcribed into an RNA that becomes called messenger RNA, mRNA, because it's carrying the messenger message to the ribosome. Okay? And so the MyoD protein is expressed when the MyoD gene gets activated. And that's the first step in our story. And there must be some transcription factors that activate the MyoD gene. And we're just not talking about them because it's just becomes, lots of these gene expression stories could be pretty complicated because I mentioned there will be multiple enhancers. And so it becomes more of an extended story. What we've done is, I said there's like a series of dominoes falling, and it's a series of gene expression falling where the cell was originally, it was a fertilized egg, and this cell, it goes on and divides and stuff, and eventually is going to become a muscle. But it's not there yet. And as it kind of moves toward its eventual fate, it's changing gene expression. So these dominoes are falling. And we can just pick 3 in a row that would be a simple, memorable story. And that's what we do. We're just going into the overall timeline and saying we're starting with, MyoD gets activated. And we say, we know why and how, but we're not learning that in this course. We're saying MyoD gets activated, and it kind of immediately binds to myogenin and activates it, and then myogenin gets expressed, and it kind of immediately binds to muscle target genes, and we start making muscle proteins. Yep. So that's always comes up. You're like, well, how did that MyoD get there? And the answer is it's a gene. It got activated in the same way, but a little bit more complicated and less sort of satisfying to start a course with. Yep. More question? Yes.
So for myoblast, I'm just confused to its function. You said it's a precursor to muscle cell, but it's not myogenin that activates it. So what does that actually mean?
Yeah, so that's a great question because it goes back to that kind of car versus tire question. Really, we're talking about the cell, and the cell is a myoblast, and a myoblast is a precursor to a muscle. And so that means that really the only thing that myoblast can do is become a muscle. And in the videos, there's an example of another kind of precursor cell. It's a chondroblast, and it's a precursor to cartilage cell. And that's what's happening as our bodies develop in embryos, right? Is that we get a bunch of cells being made, and they have to start preparing to be different tissues. So we have precursor to brain cells and liver cells and so forth. So it's the cell. The myoblast is the name of the cell, and it's a muscle precursor cell. It's not a chondroblast, it's a myoblast, like that. And then down at the gene level is where we talk about myogenin. Myogenin is a gene, MyoD is a gene, and those genes are found in every cell in every human all the time. So they're always there, and we're going to activate them to make a muscle. And one reason we talk about this little series is because you can understand, for example, that when a person is pregnant, early in the pregnancy, there is no heartbeat. There are no muscles. There are just cells that are going to be muscles. The cells already have the genes to make all the muscles because that's what we inherited from our parents. But they haven't made them yet. And so this is just a nice little story of how a cell with all the genes can activate expression to become a specific tissue. And it's a good example because, muscles have a lot of proteins in them. The striated muscle fiber that's doing the contraction of any movement we do is because muscle proteins are sliding past each other. So they're proteins, so they must have corresponding genes called actin, myosin, titin, so forth, like that. And they're not being expressed yet. So how do they get expressed? And the answer is, well, there's got to be a transcription factor, and it's myogenin that binds it to them. And myogenin protein is got to get made. So there's a myogenin gene. How did that get made? So we talk about it like that. Yep. Yep. Other questions people have? Yep.
Is methylation the only thing that turns off a gene completely?
No, there's lots of things that that regulate a gene. And so in one of the videos, they talk about DNA methylation and how when the muscle differentiates, it still has all the genes. Our muscle tissue have all the genes. Our muscle tissue has the insulin gene, but it's never going to use them. And so it's really common at that point that a cell will start methylating, and that means to add little methyl group, chemical group, covalently bonded to some of the cysteines in the DNA sequence. And in that sense, our DNA sequence is like an A, G, C, T, and it's actually kind of has a fifth letter where some of those C's can get modified, they can get methylated, and they become methyl C's. And the function of DNA methylation seems to always be to repress gene expression. And we'll see lots of examples where the DNA is not methylated, but the gene is being repressed in another way. Yep, there's lots of ways to repress gene expression. We'll talk about one today, I bet. Yep. Yep.
What's the reason that we can't take stem cells in the lab and then make them differentiate into like a red blood cell for transfusion purposes?
So what's the reason that we can't take stem cells in a lab and differentiate them into red blood cells? And so we can. But what you really want to ask about is to get them to differentiate into like functional replaceable tissue. So a good question would be to turn our attention to muscle. We can easily make muscles in a dish, muscle cells, making expressing the muscle genes, but it's hard to take those stem cells and get them to actually help build muscle in a body for a bunch of reasons. And it's kind of like, they really evolved to build muscles in an embryo and fetus, and now this is an adult, and so it's kind of like a different microenvironment around the tissues in those cells. And that's a super significant question, and scientists are making really dramatic advances in that. So, you say we can't, but another person say we can. And for us today, we just say, yeah, we can, but what really is hard to do is to get that to make tissue like functional in an injured person. Yep. Yep. Other questions people have about anything? Yep.
There was a part in the book, and I think they were comparing eukaryotes and bacteria.
Yep.
And they said their genomes were mosaics of one another. What does that term mean? And is that evidence that both of them came from a common ancestor?
Yeah, so that comes up in chapter 1, and so chapter 1's kind of ranging around and doing the intro of everything. And on our planet, there are really, three big categories of life, and they are eukaryotic cells, prokaryotic cells, and archaea. And the evidence is really strong that the evolution of eukaryotes involved a symbiotic relationship between cells, where one simple prokaryote was absorbed into archaea, and that became a eukaryote. And so it's interesting because the reason people talk about that is it really looks like that's how the mitochondria and the chloroplasts were formed in long time ago. And there's no fossils of that, but there's curious, interesting DNA information, and then also some of the functional proteins and stuff. And so just to talk about that a little bit, it's curious and interesting to see that those two organelles have ribosomes in them. Why do they have ribosomes, And, that kind of hints at maybe they used to live separately, because, you only need ribosomes if you're going to be a cell that needs to make protein. And then those two organelles also have DNA. There's a mitochondrial genome and a chloroplast genome. And so what's up with that? And so it's called the endosymbiotic theory, and it's just based on that, it's a lot of dramatic evidence that they related. And so the mosaicism, so a mosaic means a kind of a hybrid detail. And so there's a mosaic feature in modern eukaryotic cells between, say, the mitochondrial genome in the mitochondria and the nuclear genome in the nucleus. And the mosaicism is that some of the proteins that are needed in the mitochondria are made by the mitochondrial genome, and that makes sense that maybe it used to be a free-living bacteria and it got absorbed. And but others of those mitochondrial proteins are actually made by the nuclear genome. So part of that endosymbiotic theory involves the hypothesis that some of the initially all mitochondrial full genome, some of those genes actually somehow migrated or moved over to the nuclear genome. That's why they talk about mosaic genome, because it's kind of this curious mixture. And all that happened a long time ago, and so we don't know, and we will never really know, but lots and lots of really strong evidence points to that, and so people have really definitely concluded that that's what happened. Yeah? Yeah, so that's some chapter 1 stuff. Yeah. And yeah, we can do that. Remember, I always bring my book and you can just turn to a page and say, what do we need to know off this page? That's a great question. Yep. Other stuff people want to know? Yep.
Chapter 1 spends a lot of time talking about the length of the various genomes of these different model organisms. What's the significance of the length of the genome? Why are they really excited about the number of base pairs in each genome?
Yeah, so there's there's no real dramatic need to know that for our course. So that's the first question is, do you need to know the length? And no, we don't need to know the length. But it's good to know just in a kind of as you're learning way to kind of appreciate the level of how much of the DNA sequence is there involved in this. Yeah, and so that's why it's showing it there. Then there's just some curious surprises that are kind of historic. And so, for a student who's learning in the course today, there's really not too much interest in the history of the science, but there are some things in the history of the science where you're just like, oh my God, right? And so here's an example. 25 years ago, scientists were working hard to get the DNA sequence of the first whole human genome, and it was 3 billion nucleotides long. And I talked a little bit on another day about they didn't start sequencing any DNA on our planet until the 1970s, and then they increased slowly. So in the 1990s is when the United States government said, we're going to have a kind of a moonshot type effort, not to just cure cancer, but also to sequence the human genome, and it's going to cost billions of dollars, but here we go. And so they're working on that, and part of the process of getting there was, let's sequence some shorter genomes, get those done, like some bacteria, so those are getting done, and then some simpler eukaryotes. And so fruit flies got sequenced because the fruit fly, geneticists and molecular biologists, they're like, we can get this done because it's much shorter. And so just back to that history, and it was just 25 years ago, well, the fruit fly genome gets done, and they already had a lot of genetic information. So it's one thing to say, yeah, we know all the AGCTs. It's another to say we've actually labeled the genome with where the genes are. And that's called annotation, where you actually start labeling it. It's not just the letters, but you're saying like, yeah, this is where that gene is, that's where that gene is. And that's a big, big deal. And fruit fly geneticists were already weirdos kind of teaming up. So they just teamed up and they had a really good genome. Yeah. And so they said, oh, yeah, so the fruit fly has 14,600 genes, like that. And it was a ways before they're going to get the human genome done. And so immediately, for a few years, 25 years ago, scientists like me started to say, well, I guess the human genome must have 100,000 genes. Because if a stupid, yucky, little gross fruit fly, hi Alex used to work in Ken's lab on fruit flies, but little pest, right? Fruit flies, and if they got 14,000, and the yeast that causes bread to rise, it's got about 8,000. Humans are cool. We gotta have 100,000, and we have about 20,000. So the genome length is part of that, like kind of a nod to that reality, where there's some big surprises in it. Another big surprise is there are plenty of other organisms that have much bigger genomes than us. Like there's some salamanders that have genomes that are like 10 times bigger than ours, you know? And you're like, what's up with that?
Are
Yep.
those the ones with the gene duplication from the hybridization events? Like we have them around here, like the blue spotted salamander.
Yeah, I don't know the background, but there's some surprises where you're like, must be more, genome must be better, right? And more genes must be better. And there's some cool explanations for why we could make a human with only, several thousand more genes than a fruit fly, And the explanations are cool like that, but we're not going to talk about them because we have to focus on today's learning. Yep. Yeah. Other issues people have? Yep.
I just had a quick question.
Sorry for teasing you, Alex.
That's okay, I can see. When you talked in the video about DNA methylation and queen ants, you just briefly mentioned that they become that way because of the DNA methylation pattern, and it's caused by the food that they're fed as a larva. Why then is there only like one queen ant? Like why aren't there more queen ants?
Yep. Yeah. So a queen ant is an identical twin to the worker ants. They look really different. A queen ant is huge and is laying a bunch of eggs. The worker ants are small and they don't lay any eggs. But they're identical twins. So what happened to them? They have the same genes, same alleles, right? And the difference is that when they were larvae, the queen ant was fed this stuff by the workers that's called royal jelly. And it's just food that the other ants chewed up and spit out. And when they spit it out, it included a hormone. And the hormone is what causes the queen to form. And so the answer to your question is they just have a little teamwork plan to just make one queen. We're going to make one queen. It looks like we need a new queen, so let's get that royal jelly going. Chew it up. Come on, spit it out. Yep. Yeah. Yeah. And that's just an example because lots of people, lots of students today are very interested in DNA methylation and epigenetics. And one of the reasons is because it's relatively recently that scientists could rapidly analyze the pattern of DNA methylation. And so there's lots of discoveries and lots of really significant applications like that of like, oh yeah, the difference between a queen and a worker is just DNA methylation entirely. It's the same genome, And you're like, how'd that happen? And they are like, well, they fed them some royal jelly. And they call it royal jelly because they've known about it for years, right? Little ant keepers and beekeepers are the same story. Is they make the queen by feeding them this royal jelly. And it's just chewed up food. It's just the regular, ant food, bee food. And some of the workers chew it up and they feed it to this one larvae. And they like, hey, you're going to be great. Someday, you're going to be like the equivalent of like Simba kind of thing, It's like, they should have, right? They should make a Disney movie of that where there's a little bee larva hanging out looking really simple. And they're like, come on, sing a song about how much you want to be queen. Yeah.
We should sell that to Disney.
We should sell it, or we should just make it. Come on, you guys. We could.
What Shakespeare play would it be based on?
King Lear. I don't know. Yeah. But anyway, okay. Other questions people have. All right. And so you can interrupt with questions anytime, right? And they can be anything, right? And so those are good examples of that. So let us now. We're going to do this. So we're going to do this thing, right? And so let me start out before you start drawing. And if you haven't gotten drawing stuff yet up here, you can just get up and start getting a little bit noisy. But let me just review this thing that we talked about last time. Let's go, little drawing tablet. Uh-oh. Oh, my drawing tablet is somehow frozen up, which is not good. Yep. So we'll just try and plug this in. Okay. So on Tuesday, the big take-home thing was that we labeled the names of each gene. And so this is a DNA sequence, and you know, we talked a minute ago about the length of genomes, length of genes. We've really zoomed out here. This is a piece of DNA, and most genes in our bodies are tens of thousands of letters, nucleotides long. So we've zoomed out, so we're not writing down the AGCT. And all the DNA is that AGCT pattern. But you know that in a book, it's just letters, but the letter pattern is organized into words and sentences and paragraphs and chapters. And so when we actually look at the function of the DNA sequence, we find places that are like a word, a functional thing, right? In words, in the English language, between the words there are spaces. In the DNA, it's physically connected. There's no such thing as a space. So in the chain of nucleotide letters, there are gaps of just useless information. And so when we look along the length of the DNA, there are big regions that seem to be just garbage, and then there's a patch that's significant, and it's just like the AGCT letters. And so that's what we've drawn here, and it's still just the DNA, and it's just the letters, but we've studied it, and we know that there are these regions. And this first region is the transcription unit, and again, it's just AGCT letters. But we studied it, and we've seen that RNA polymerase lands there and transcribes that. So the transcription unit is the region that gets transcribed. And around it, that's just some garbage spacing letters. You know, they don't mean anything. Yeah? And then there's a promoter, there's an enhancer. And those are the main things. It's really important to appreciate. Those are DNA. They're just DNA letters, but they're organized like a word or a sentence into a functional series of those letters. And I will just draw it up like that. I'll just draw that kind of abbreviation, but you need to be able to label that and explain it, and you'll see today in our drawing we'll manipulate it because it's very physical. It's very physical, and it's very matter of fact, and it's just a gene. I just sketched out a gene, and again, I do it fast because I want to keep rolling, but you need to be able to label it out and explain those parts, right? So transcription unit, and that's going to be the output. That's the functional output part of the gene, and so that's the part that's going to get transcribed. So it's going to get transcribed like that, boom, and then going to get transcribed again and again and again. So I have a bunch of these RNAs, and if it's a coding gene, it's going to have a coding region in the transcription unit. The coding region is going to have codons that the mRNA is going to be used by the ribosome to code for the protein product. So that's just a summary. We did that on Monday, and we were just trying to organize that and understand that. Then, on the enhancer, we wanted to write this. Well, you know, the MyoD protein, it's a protein, and it binds here, and that's the switch. So a key concept in turning a gene on and off is the gene is not changing. The gene DNA is stable. The dynamic, the changing part of the story is the transcription factors that are binding, and that's a huge concept. And water is H2O. We just need to understand that fully. And if you need to watch the videos again and again and draw it out again and again or make models of it, yeah, it's just a matter of fact thing. Yep.
The enhancer gets bent to bind to the transcription factor?
That's a good question. The answer gets bent to bind to the transcription unit, and you'll see in the clay model videos, I always draw it like it's a curved gene. That's because the DNA is floppy. It's not cooked spaghetti. It's a lot of times it's drawn out like it is straight, like a stick, like uncooked spaghetti, but it's cooked spaghetti. It's floppy. And we'll see that there's a reason for that floppiness, and we'll learn about that in chapter 7, which is we're coming up. Like next week, we'll answer a question about how the DNA, how the transcription factor functions. Yep. And we'll add a little bit right now. Okay? So people still really like to call that DNA where the enhancer binds, they like to call it the enhancer. And that's because historically, people found that was the big switch. If they deleted that, the gene didn't turn on. It was the big switch to turn the gene on. And again, the enhancer's not changing, it's just DNA letters, but it's a landing site for the big transcription factor, MyoD. Like that, it's a landing site for the important transcription factor. People like to call it an enhancer because for the first genes they studied, it really enhanced. It increased the synthesis. It increased the RNA production. So it enhanced gene expression. But then there's reasons why they want to call it enhancer still, and that is because the enhancer was a big surprise. Because as scientists study it, they found this. There are 3 features, let's see, 2 features of an enhancer that we need to know about. And one is that it acts at a distance. So notice that there's a gap here. That's the distance. The enhancer can be far away from the transcription unit. And so that's the first thing, is it acts at a distance, and that's just a fact about an enhancer. And your question is, how can it act at a distance? And the answer is, well, the best evidence is the DNA is floppy and it's going to bend to get this close to that.
Because you said that it would tether, like the
Yep.
MyoD would tether, so I just didn't know if it was like an actual connection, and then like once it was pulled away, that's when it would stop the transcription.
So the way I describe it is, do you imagine that the transcription factor is a bitey dog, and it's on a leash? And the leash could be pretty long, but if you're holding the leash, you're going to get bit really quickly. If the enhancer DNA is mutated, so the enhancer can't bite, the bitey dog is just running around the building, and we're not going to get bit. It's there, but we're not going to get bit, because, you know, it's really unlikely. But as soon as you put it on a leash,
She doesn't bite.
I got bit by a dog this, like 2 days ago. Yeah.
Wasn't mine.
Look at that. And, but it's just tethering it. So these are like garbage letters here, but they are physically tethering it to a leash. So it's in this neighborhood, right? You're holding it close to you, and the transcription factor is tethered, like that.
Or like those run leads things.
Well, it's just flexible tether. One phrase, one term that biologists like to use that's a satisfying term is this. So it goes like this. So in the cell, that transcription factor is made a bunch. So there are tens of thousands of molecules in the cell. So there is a molarity, right? There's a concentration of those. And the concentration of the dog in the building is 1 dog per building. That's the molarity. And it's low. And what is happening here is this tethering, scientists will say, increases the local molar concentration. So you still just have 1 dog in the building, but it's now here. The concentration went way up. And it's still running around, but it's way high. And it's gonna function, you know, because it's here now. But it's kind of like a molarity concept. It's kind of a concentration concept. The concentration was really low. It was there was lots of it in the cell, but it was all over the cell. And you just tethered it, so you just located it here. And the first thing is, the tether could be different lengths, and it doesn't really matter. You know, double the length, who cares, right? Like that. And then the second thing is, you can take the little enhancer and you can just flip it around, and it works the same. So you could just rotate it 180 degrees. So it's not a directional thing. It's not saying, go that way, you know, there it is, there's the target. It's reaching out in both directions. And that makes sense too, right? If you took dog's collar on upside down, it's still going to be, you know, tethered. So either way you tether it, there it is, right? Like that. And that has implications for what the transcription vector is doing. Because scientists first thought, maybe it grabs hold here, and then it slides down the DNA to get to the target. But if you can turn it around, it can't be that, because it'd be sliding the wrong way. See? So that are a couple of reasons why people are like, I like that word enhancer, because it kind of underlines the curious nature of it. And in chapter 7, we'll talk about what the transcription factors do to regulate transcription. So we'll get there to add more detail to that. But that's a good place to talk about the enhancer. Okay? So the second thing is acts in either orientation. And you see how cool this is? You know, like, you know, you're thinking about it and you're like, maybe it just grabs hold there and slides down the DNA. We know it has to be connected, so maybe it's not so much a leash thing as just a pathway. But then you say, no, we just turn it around and it works the same. And the third thing, which isn't really a third thing, it's really like the second part of the first thing, is the third thing is that it even acts downstream of the transcription unit. So, you know, we always draw the enhancer up here, but there's some genes where they have an enhancer over there. And that goes back to the tether, if you know, well, the tether, there's a tether, and that's also a tether. That's holding it close. And so, but it was another curiosity, because again, how does it know to activate that gene if it works great when it's downstream of that gene transcription unit? You know? And it's just, you know, and scientists have some good comments about that now, but it was big news, you know, and so people like 20 years ago, like, whoa, enhancer's a cool name, I like it, you know? And so I don't know for sure, but I think in most courses, most teachers would still call it, it's an enhancer. And remember, and we'll learn today if we have time, or next Tuesday, we'll learn about examples that are definitely not enhancers, they're repressors. Yep, okay, any questions? Yep.
The repressor and the enhancer.
So I saw you getting ready to ask a question, and this is how you ask a question. What was the subject line for your email, Joseph?
Oh, what's up, loser?
Yeah. What's up, loser? That's, you know, you can raise your hand, or you can just be like, hey, what's up, loser? Yeah. Yeah. Go ahead, try it.
Is an enhancer and a promoter
B minus. Huh?
a distinct? Are those distinct binding regions?
Yes.
Or
Yes.
Can we
There's the enhancer. There's the promoter.
I'm sorry. The enhancer and the repressor. Are those distinct binding regions, or can we say that different proteins have repressive tendencies when they bind to the
So
multi-binding
either
unit
today,
of
but
the
now
enhancer?
probably more likely Tuesday, we're going to talk about a transcription factor that's called the estrogen receptor. And you already know, oh, that must have something to do with the hormone estrogen. And we'll focus on that, but we could just as easily pick on the testosterone receptor. Those are the receptor protein that binds those hormones and causes the cell to respond to those hormones. So every hormone has a protein receptor, and the receptor activates a response. So we're talking about that. And the estrogen receptor and testosterone receptors are transcription factors. And in the absence, and I'll just draw, and again, we'll just focus on the estrogen receptor, but we'll get to that. We'll want to talk about that example because it's just satisfying, and we might also just get to it now. So there's the estrogen receptor, and people like to just abbreviate it ER, estrogen receptor, right? And just to mention it, right, all vertebrates have estrogen and testosterone, and they have those as the sex-determining hormone. All vertebrates. So all fish, you know, like that. All vertebrates have those as the sex-determining hormones. And they all function during sexual maturation. So puberty. All vertebrates have a non-reproductive juvenile and a reproductive adult, and these hormones cause the reproductive maturation. So a key concept is that in humans, these hormone receptors are here in children, but the corresponding hormone is not. So in juvenile females of any vertebrate, girls, human, the estrogen receptor is there, and it is repressing gene expression. And then, when the estrogen hormone shows up during puberty, it activates gene expression. Same transcription factor that's changed its function 180 degrees when the hormone shows up. It's a repressor in juveniles and an activator in adults and teens, okay? Here's something else that you know about our species that, you know, you don't take to be surprising, but it's curious because it wouldn't necessarily be true. You know that an adult male could get shots of estrogen and develop secondary female sex characteristics because the estrogen receptor is present in every one of us, and the estrogen receptor is present, and it's repressing female-specific genes in juvenile girls and all males. And that's why shots of estrogen would cause some sex characteristic changes. And testosterone does the corresponding opposite. Yeah?
I know estrogen is used in like cancer therapy. How does that work?
We'll get to that. So estrogen is used in some cancer therapy. And especially we'll talk about breast cancer. And there are some breast cancers that are said to be ER positive. And there's some breast cancers that are ER negative. And they're referring to whether the breast cancer cells are expressing the estrogen receptor or not. And the ones that are estrogen receptor are sensitive to a chemotherapeutic chemical that inhibits them. And so for people who have breast cancer, the first thing they always do is they check, is this breast cancer ER positive? About 70% of all breast cancers are ER positive, or is this one ER negative? And they're saying, if it's ER positive, we can use the drug. If it's ER negative, the drug will be useless. And that's a really good example of getting ahead to like chapter 20, right? We're heading toward that. And it's really satisfying because you know there's situations, right, where here your neighbor had breast cancer and they had the drug and it helped. And then you go to the doctor with your own treatment, your own diagnosis, and you're like, well, my neighbor got this. I'm looking forward to getting that. And they're like, we're not going to give it to you. And they're like, wait, what? And you're like, you're ER negative, so no. And they're like, come on, let's just try it. And they're like, no, we don't have to. And that's this kind of thing, right, with medicine, right? You talk to medical doctors where you're like, maybe it would be a miracle and it'll work. And they'll be like, no, it's not going to work, right? Because it's molecular biologists really talking through the doctor to you and just being like, yeah, it's not going to work. Because the way the drug works is it binds to that. And if you don't even have the protein being made in the breast cancer cell, it's not going to help. Yep?
The same thing in men. Like more specifically, because my grandfather has stage 4 prostate cancer, so they've used estrogen therapy.
Yeah, so then there's going to be a bunch of stuff that's like medical questions, and I'm like, I don't know. Yeah, why they would use estrogen therapy on prostate cancer, I don't know. And it doesn't mean I'm a clueless loser. Thanks, Joseph, for that. It just means that, I don't know. And I don't want to speculate, right? It's a big mistake to just be like, because a lot of times I'll have an idea. I'd be like, I have a suspicion. But it's not, that's not good for me to stand up here and go, well, as a matter of fact, I think, it's probably not the same thing. Yeah, no, I'm not going to do that. So, but we'll get to that. And it's really, anytime we start talking about a cancer story, it's really difficult. And it's difficult because, there are people involved, but also it's difficult because we'll see that the causes of cancer down at the molecular level are always just so unlucky. Just as such an unlucky thing happened, 1 in a billion thing, it happened. And it's just like, I can see why that's bad, and it is. It's just bad. That's a bad situation for the cells, Yeah, so we'll get there. But so we'll talk more about that example. We'll talk more about the enhancer. But just saying, to start with, last on Tuesday, we drew just a map of a gene. And it's a universal map of a gene. And its transcription unit, that's going to be transcribed into RNA and be the output. The primary output of any gene is RNA. The secondary output could be a protein, if it's a coding gene. And then these regulatory regions. So then the second thing, it was the only other thing we accomplished during our drawing assignment on Tuesday, and we won't be able to get to maybe today, because we have our quiz, right? So we're not going to get any drawing done today, right? Yeah, but it's like, this is like it's amazing for me to be saying, this is prerequisite knowledge, that we need to walk in with, but you don't have it, and I know that, so it's amazing that we got to get this down because it's really foundational. So we all got to understand it. So the second thing is this that we accomplished on Tuesday, and we really need to know because. Like when you're drawing and you're working, it's just fun. You're in kindergarten. You're like, I want a juice box, like that, But we're actually doing some learning. So it is a challenge to practice this drawing and stuff because it doesn't feel like learning. But we're trying to spread it out. We're trying to build a resource that will help us learn. So the second thing is we talked about how random mutations could affect this gene, the myogenin gene. And the mutations could be either here or up in here. They could either be in the transcription unit or this regulatory region. So we could get a null allele by destroying a protein by making a change in the amino acid sequence so it doesn't work. Or make a mutation up here in the enhancer and it doesn't work. Gene doesn't work. And but then we said, the phenotype of those, if they were, null alleles, would be no muscles, right? So that's the phenotype from those genetic mutations, And then we said, there's also that myogenin gene, and that could have mutations, and if it's defective, or the MyoD gene, if it's defective, the phenotype is indistinguishable, same phenotype, no muscles. And those could be here or here, either in the transcription unit. And what's happening there is somehow we either make a defective MyoD or we make no MyoD. And even if we have a wild type normal myogenin, we just don't get muscles. And so, many years ago, when geneticists made, found mutations in spontaneous mutations in nature or induced mutations in lab, they map them. And they're like, these mutations are all really close to each other. And then those mutations are super far away. They're on another chromosome. And they didn't understand why those could give rise to the same phenotype. But they said, these are physically connected. These parts, they're like tethered together. And so we're going to call these cis, and these are not. And we're going to call these trans. And that's a term, you learn that term in chemistry, cis and trans, like that. And, I think they're saying here kind of probably the same root use of cis is sort of next to each other and trans is sort of across from each other. So, but the key thing here is it's not chemistry, it's biology, and cis means physically connected to each other and trans not connected. Cis is helping us define a gene. These are cis mutations that affect that, and that's a gene. That must be a different gene. And so that's where the word transcription factor probably helps come from, because it's acting in trans. And one of the drawings that we'll do next Tuesday, again, same thing, but we'll just try to explore and appreciate that a little bit, that idea that it's acting in trans. Because in our DNA, we have genes, and the genes can get turned on and off, but they get turned on and off by a different genetic region. So we inherit from our parents both the gene and the capacity to turn it on and off, right? Because the DNA has to hold all this information. It has to give you all the genes you need to be a kid and then go through puberty, several years later. So there have to be delays built into the use of the DNA, like timing, spatial, and amount delays. And the regulatory region does that, but the regulatory region isn't changing. The regulatory region is just a landing spot for these actors that come in trans. Yep. And again, this is just universal. This is how life works on our planet. In every cell on our planet, DNA is the primary heritable molecule, and the organism gets it when the cells, sperm fertilize the egg. There's the DNA information that organism is going to have for the rest of their life, and then it gets used over time because it has both the information and it also has the regulation of the information built into the whole DNA sequence. So again, very physical and matter of fact, and from this we call this now. We can call it the regulatory region, that's great, but the superficial final term for it is the cis regulatory region, because it is acting in cis. Yep?
So if there was a mutation on the next, domino, the next gene, after myogen, then would that be the cis too, and the one before would be?
No, so it's always trans if it's a different gene. So when this turns on muscle genes, those are also, they're trans, because it's a different gene. Yep.
Okay.
Cis means same gene. Same stretch of DNA, same gene.
Dave, does this imply that the entire MyoD region is the transregulatory region of the myogenin gene?
So that's a good question, and people would say kind of yeah, but remember that we can move the enhancer around and it still functions fine. So, you know, when we say the whole region is functional, because most of the region is just the leash. So it's functional because it's physically connecting it, but the letters don't matter. You know, we could completely change the letters around there, but it's still tethering it. It's still tethering it means keeping it in cis. If we make a break here, it can't work. You know, if we cut the leash, the local molarity goes away because the dog's running around, you know, and just takes off. It's got to stay tethered, so that's the cis concept. They're physically connected to the same strand of DNA. They have to be connected to do their job. To have that, those, you know, enhancer has to be connected near, you know, in the vicinity of the transcription unit, but it could be downstream, it could be turned around, it could be longer, shorter. So yeah, people would say, yep, that whole region is the functional region, even though there's big chunks of the region that don't matter what the sequence is, it's just that it's connected.
And then how far back do we get to go, right? So myogenin has a transregulatory region which consists of the MyoD gene, which has transregulatory regions consisting of further upstream pathways.
Sure.
And so would we describe all of those as transregulatory regions for the myogenin gene?
Yeah, everything that's not that gene is trans. Cis would be the gene, and everything downstream or upstream of it would be in trans. And those are genetic terms, but we understand them now physically and biochemical and, you know, microbiology terms. Yep. So cis regulatory region. So we'll call it that. So every gene is composed of two parts. Oh, and let's get this definition in before you take the quiz, because our beepers are going to go off, and so let's make sure we're solid on this, because we're kind of upgrading our definition of gene. And we'll talk about this more on Tuesday, but gene is the name for the field that studies genetics. And it turns out during the genetics history, it's only been practiced for a little over 100 years. So there's a field of study, genetics has only been, you know, a topic for about 100 years. And for the first half of the history of the field of genetics, no one knew what a gene was. So it was a theory. And then it was discovered that it was part of DNA. And then it became the opposite of a theory. It became a fact, a physical fact. So lots of people have a hard time defining gene. Even, you know, professors of genetics are like, oh, I don't know. If you look at the Wikipedia page of the word gene, it's just the world's crappiest definition. You know? And it's just because genetics is a young field, and there's still some people that think it's somewhat theoretical, but it's not. So when we say a definition of a gene, we're the molecular biologists who discovered it. And so, yeah, duh, you know, like that, right? It's like that. Because a lot of molecular biology, it's like car mechanics, where the car mechanic is like my furnace is broken, and I called those people, right? They're furnace mechanics. The first thing they say is, do you have oil in your tank? Right? Because they run into that, right? Where you just need oil, and it's $220 for us to show up and tell you that, you know? Right? Right? So it's like that, right? So for a definition of gene, it's just dead easy for molecular biologists, because the first thing you say is it's physical. A gene is physical. And because of the first half of the history of genetics, it wasn't physical. We're still hammering that home. And we're hammering it home to everyone, especially students that are learning it. Oh yeah, it's very physical. You know, that's why I made it a clay model. That's a universal example of a gene. Every gene is very physical, and it's composed of DNA. On our planet, that's it, right? And then it's two parts. It's made of DNA, and there are these two parts always. And you could call it, you know, the two parts are the functional part, it's going to make a product, and the regulatory part. So we're fancy now. We could say two parts, and there's always a transcription unit and a cis regulatory region. Sweet. And then we want to tag in there, and we'll get around to this later in the semester, but we also want to say, and it affects the phenotype. There's some genes you might have heard of called pseudogenes, and they're just old-time genes that are destroyed and they're fading away in our genome. We have hundreds of them. And they're just all broken down and fading away, but it takes a long time for them to completely disappear because you have to have mutations just randomly change all the letters. So those aren't real genes, they're pseudogenes. So a pseudogene must affect the phenotype. And there are other reasons why we want to put that in there. But yeah, so that's a question on the quiz that we have to take now. How come nobody's beeper's going off?
It's just 30 right now.
Yeah, I know, but when do you think your phone's beeper goes off? Doesn't it go off then? Okay, so put your junk away, and when you're done, handing it up here, and then if you have some pens that don't, we didn't even draw. Oh my God. Right? How sad is that? So your quiz 1 is up here, folded over so no one can see your score, and it's stacked in alphabetical order by your last name. Okay? And to make it a little bit easier, I'll put the students with the name A through M right here where the quizzes are, and N through Z right where you're putting away your. Yeah? So make sure you just take one, right? They can get kind of stuck together. Then I'll hand out these quizzes, and when you're done, hand them in, and then remember there'll be that quiz 1 answer key. You gotta watch that, and then we'll do the drawing assignment on Tuesday. So. Right? And so I as a kid grew up working on a farm town setting like that, and when I ran into this topic in college, I was like, oh my God, this is the coolest stuff. I just want to have this information. Because it's true, it's just true, you know, like that. And we'll learn it, we'll be careful to, set it up so everybody is keeping up and understanding it. That's great.